Hello. Hello. Hello.
Yes, sir.
Just 1 minute, Amma.
Okay.
Yes, so audible.
Yes, sir.
So last class, what we are completed? Hello. Only 2 members is there. Hello. Please respond, Sujata and Ramya. Only
Yes, sir.
two members are present.
Yes, sir.
Yes. So today, last class. So we discussed about general characters, diversity of taxonomy, and utilization of fungi. Yeah.
Yes, sir.
Before going that one, so how many types of reproduction in fungi? Tell me here how many types of
Vegetative, sir. Sexual and asexual.
asexual reproduction systems.
Yes, sir.
Okay. So it is already completed in the last classes.
Yes, sir.
So before going that one, just I'm giving the introduction about once again what is the fungi. The fungi are achlorophylls. Already you know very well. The fungi, it is a saprophytic, and another one that is often parasitic. With it is in unicellular. The more typically filamentous, all the fungi, it is a filamentous type. The vegetative structures, so that is either sinusitic or which one that is often saptate. So this is generally the fungi surrounded by cell wall composed mainly that is a chitin and so that is other polysaccharides. The fungi propagation fungi by the which one that is often by spores, different type of the spores propagation by spores and zygospores. So many types is there. So already you know very well. By spores and reproductions. Okay, reproduction normally by both asexual and sexual. The most of the fungi, their nutrition, absorption of nutrient transported across the cytoplasmic membranes into cell. And also one more important, the fungi, they play an important role in the biosphere. The fungi is a decomposers where the organic matters. They are edible and also used. The fungi, it is edible. They are using in where is the most the food industries and beverages industries. So and they are most important in the industrial. The source that is of antibiotics, vitamins, and other enzymes. Okay, the fungi produce all these things. The see here the nature they exist of pathogens, that is often saprophytes, symbiotics, and other one that is in predators. Other one that is often predators. They occur derives in habitats such as soil, water, decomposes, and they are also extreme environments. So this is a little bit introduction about the fungi. And the next one, the fungal taxonomy.
Excuse me.
Yes,
Page
tell me.
number, please. Page number, please.
This is of in all introductions. Already you are completed. This I am giving the brief. So you see here the second one that is in the fungal taxonomy. So you know the what is fungal taxonomy? The fungus is divided mainly. How many classes? Tell me here. Fungal taxonomy. How many classes? Mainly.
3.
Yes?
5 classes, sir.
No. The mainly fungus is divided. 2 classes.
3. You might continue.
Yes. Please tell me. Sujata. Tell me, it is very easy.
Masticomycotina, zygomycotina, ascomycotina, parasitic.
Before that one, before that one, entire the fungus is divided mainly 2 categories. Okay, entire the fungus mainly is divided in 2 categories.
Mycotina.
Mycotina.
Yes, other one. Another one?
Mycotina.
Yes. Okay, so we call that as a Myxomycota and Eumycota. Okay, so Myxomycota means, what is the meaning of Myxomycota?
Absence of nucleus.
No, it's wrong. You see, instead of cell wall, there we are looking in only plasmodium structures. So we call that is in Myxomycota. So second one that is of in plasmodium or pseudoplasmodium is absent. There is of an assimilative filamentous is there, that is in Eumycota. Once again, I am telling here, see entire the fungi, the mainly divided into categories. One, it is in Myxomycota. Second one, that is in Eumycota. Okay. So Myxomycota means the cell wall. The cell wall is made up of plasmodium. Okay. So next to Eumycota, you see the assimilative phase, typically that is a filamentous type. So that is of Eumycota. So already completed in the last classes. So once again, I am repeat this all this one. See, first one is Myxomycota, second that is of Eumycota. So Myxomycota, we call that is in false fungi. Why you are saying the false fungi? Absence of cell wall. That cell wall absence. So we call that is in Myxomycota. Second one is Eumycota. Eumycota means true cell wall is present, is there, true cell wall. That is Eumycota. Yes, now this one, this Eumycota is divided into 5 divisions. Now tell me please what the first one is.
Mastigomycota.
Very good. Second one is
Zygomycota.
yes. Ascomycotina.
Ascomycota.
Basidiomycotina.
Basidiomycota. Deuteromycota.
Yes. This 5 belongs to Eumycota. Please remember. This 5 belongs to Eumycota. First one, Myxomycota or Mycota. That is a false fungi. Only one answer is there. Absence of. So there is no true cell wall. That is an option. So here we are looking the true cell wall. That is in Eumycota. Already we are completing this one. Once again, so I'm giving brief section. So here now you see the fungi. It is a cosmopolitan distributions. Okay, they exhibit a wide variation in their habit, morphology, and other one that's development and physiology, the biochemistry and reproduction systems. Different variation is there. So you see here the diversity, economic importance, several attempts. So made it is identified, the characterized and classify fungi. So in recent all biological literature, fungi were given the status of separate kingdom. Okay, so nomenclature fungi is strictly so governed by which in that is of in ICB and rules and regulations, International Code of Botanical Nomenclatures. And order to propose the classification based off which one that is of in phylogenetic principles. They are followed in several criteria such as a morphological, anatomical, and ultrastructures, physiological, other biochemical reproductive characters being annotated. In order it is overcome the problem studying of so where an unculturable fungus, the recent years molecular methodology such as see the fungal culture, that is a PCR technology, RAPD technology, RFLP technology. Okay, so that is of in all these the cultivation to different type of the methods they are using. Okay, so the several classified I have been proposed different type of the fungus, but now we are followed in only Ainsworth classifications. Okay, so this is a important question in your public examinations. Explain briefly the classification proposed by Ainsworth. Okay, Ainsworth classifications. And here you see that is of in RAPD. So what is the meaning of in RAPD? So this is of in RAPD means random amplified, means here this of in criteria of the classifications, how the classified the fungi. So already you know Mastigomycotina, Zygomycotina, Ascomycotina, Basidiomycotina, you know very well. So you know already Eumycota and Mycota. And in spite of, see what main criteria they have to take in. So here, mycologist means studying of the fungi scientist. We call that is in mycologist. So in all the mycologists, the types of character basis for their classifications. So different type of the sections. Number one, that is of morphological structures and anatomical structures. So morphological. What is the meaning of morphologicals? Where the external features, based on the external features. Okay. So external features means, for example, Masticomycotina. So there we are looking in flagella. So where we are looking in flagella, so that is belongs to Masticomycotina. Okay. So where the morphological features, when you are going to the classification, we call that as morphological characters. Similarly, second one, anatomical characters. Anatomical means, so now you see here two type of the fungus is there, two type of the fungus. So when I'm taking this section, so we are going to undergoing to the microscope. Based on that one, when you are going to the classification, we call that is anatomical classifications. So next one, that is of ultrastructures. Ultrastructure means, so what type of the spore shapes is there? Okay, so they are using of based on the electron microscope. Based on the electron microscope, so they are using the structures. Okay, how the formation these fungi. So based on that one, so we are going to the classification that is of ultrastructure features and their physiological characters. What type of the physiologicals means the nutrition, all things, how it is going on. So based on that one, when you are going to the classifications. So and already you know very well biochemical characters. Means the fungi produce different type of the chemicals like alkaloids and phytotaxins and everything is based on the chemicals. They are going to the classifications and reproductive organs. So next one, that is of molecular methods of identification in the fungi. Molecular methods. So molecule based on the molecules. And next one, that is of already I told in one is a PCR method. The PCR means that of polymerase chain reaction method. Second, that is of RAPD and RFLP. These three is most important in the short questions. Random amplified polymorphic DNA. Okay, DNA polymorphisms. DNA polymorphism structures based on that one. And PCR means of polymerase chain reactions. So what type of DNA structure is this? And next one, that is of restriction fragment of length polymorphisms. We call that is of RFLP. Okay, and protoplast of nuclear fusions is there. So these are all classification types. So this is of generally based on the classifications. And next, now you see here. So mycorrhiza. Already you know very well in mycorrhiza. Please tell me here what is the meaning of mycorrhiza? Mycorrhiza. MYCO, myco means fungus.
Association of fungus with higher
Very good.
plants.
In higher plants. In higher plants. Okay. So we call that is in mycorrhiza. What?
Angiosperms.
Yes. In a higher plant, that is enough. Okay. Sometimes are like in gymnosperms and also. That's why you say it is only in higher plants. The fungi association with roots. The fungi with association roots. Where it is in higher plants. Okay, that is of in mycorrhiza. Okay, mycorrhiza. So see here, the mycorrhiza or the symbiotic association between fungus and roots of higher plants. Already you know very well, there are different type of mycorrhiza with mainly two is there. One it is in ectomycorrhiza, second one that is in endomycorrhiza. Okay, ectomycorrhiza and second one is endomycorrhiza. So you see ectomycorrhiza from thick mantle on surface of the root and produce into the cortex. Okay, contingent cortex is there. In endomycorrhiza, orbuscular mycorrhiza are important in that one. Orbuscular. Okay, this is also the short question purpose. It is most important. Orbuscular mycorrhiza. So orbuscular mycorrhiza, this association fungal symbiont forms of ramified structures known as orbusculus inside the host cell. Inside. Okay. Ectomycorrhiza fungal symbionts belongs to Basidiomycotina. Generally means this mycorrhiza means it is association, symbiotic association where it is in Basidiomycotina. So whereas the orbuscular mycorrhiza fungi belong to that is of endogonales of Zygomycotina. Sometimes Basidiomycotina and other one that is of Zygomycotina. And next you see the ectomycorrhiza fungal can be isolated. So where it is in culture and organic media, the orbuscular mycorrhiza, the fungi because their obligate symbiotic nature cannot be cultured in artificials. Obligate symbiotic. These fungi cannot culture in where it is in the our laboratories. So now you see here, this mycorrhiza, it is a very much helpful to the higher plants. The mycorrhiza helps the plants of enhancing where it is in the phosphorus absorptions. Okay, so there are also the capacity absorbs of the nutrients. So drought tolerance is this. So with the transplantation shock in the resistance of heavy metal pollutes. So both type of mycorrhiza either alone or in combinations with other or beneficial organisms like it is a ribo, so that is of rhizobiums. Usually it is a biofertilizers, organic industry. Means here mycorrhiza are using, so it is a very much helpful too. So it is using to agriculture industries. Okay. So and the mycorrhiza very much helpful to the plant growth. Okay. So here this is a mycorrhiza. Is it clear now? Mycorrhiza.
Yes, sir.
Describe the importance of agriculture and forestry. The question, describe the importance of mycorrhiza. So here you give the first definition, what is mycorrhiza, and so this mycorrhiza we are using. So where the end the agriculture is absorbing minerals, proteins from all other things. So you write like this, and so already you know very well that is of an ectomycorrhiza and endomycorrhiza. Yes, so now you see here, this is also very simple, very easy. Edible mushrooms. You know the mushrooms? Tell me here, anybody.
Yes, sir.
Mushrooms.
Mushrooms.
Yes,
The
sir.
mushrooms are heavy, proteinaceous,
Food.
heavy nutrient. Okay, nutrient values is this. Okay, that is the mushrooms. So the edible fungi included mushrooms. Already you know very well. Umbrella shapes is there. Below one stalk-like structures is this. Okay.
Mm.
Voice is received. Please. So many types of mushrooms is there. So here very much nutrition values is there in that fungus, in that mushrooms. Okay. So that is often proteins is there. Second, there is of fats is there, carbohydrates, fibers, and vitamins, minerals. Okay, so many values is this. So when you are taking the mushrooms, okay. So here mushrooms cultivations. Before going that one, here these mushrooms where the puffball mushrooms is there. The puffball mushrooms are belongs to generally that is of in Basidiomycotina. Basidiomycotina. So large number of mushrooms grow is a widely. So we are looking in the nature. So generally the important where the cultivated mushroom that is of in already you know very well where it is in agaricus. You know the agaricus?
Yes.
Already you are in your first year practical classes. Okay.
Yes, sir.
Yes, so many types of mushrooms is there. So proteins or different type of the minerals is there in that mushrooms. So you see the mushrooms protein consists of all amino acids required by human body. It also contain vitamins and minerals, little for sugars, high fiber content, 90% of moisture. The mushrooms lack starch, hence suitable for diabetic patients. The most important here. And here, low sodium and high potassium ration control of hypertension. Mushrooms have immense medicinal value. So many types is there that is of. They are of hallucinogenic property, treatment of mineral, different type of the disorders. Okay, this is of into the mushrooms. So next now you see here and mushroom cultivations. Mushroom cultivations. Generally, the mushroom cultivations there are 2000 here showing now this one is visible.
Yes.
Yes.
Yes, sir. Yes, sir.
See now here this is importance of mushroom cultivations. Okay, so it is to very simple. Just I am giving in only some points. Okay, so there are near about in the mushrooms. So more than 2000 edible fungi throughout in the world. Okay, 2000. The mushrooms are fleshy, that is often macroscopic spores bearing the structure. Some fungi belongs to generally that is of Basidiomycotina group, and other one that is of where it is an Ascomycotina group. So you know what is Basidiomycotina and Ascomycotina? It belongs to Eumycota or Mycota.
Mycota, sir.
Oh my God.
Yes. Once again,
Mycota.
Eumycota. Very good. Tell me one example of Basidiomycotina. Not mushrooms, other. Other.
Rhizopus. Bread
Rhizo.
mold.
What?
Bread mold. That is.
No. That is not in Basidiomycotina.
Okay.
First year you are completed. Basidiospores.
Yes.
Yes. How many Basidiomycotina? 5 spores is there. SL spores. Pycnidiospores, SL spores, iridospores, teltospores, and Basidiospores.
Puccinia.
Very good.
Puccina.
Puccinia. Okay. But here no need of Puccinia. But the mushrooms, some of the mushrooms are belongs to Basidiomycotina group and Ascomycotina groups. Okay. Here now you see here in mushrooms, they are good source of vitamin B, folic acid, and other of blood building vitamins useful that of in anemic conditions. So they are also contain of pantothenic acid is there, where it is in B12. So many minerals and nutrients is there, where it is enough in mushroom cultivations. So, 1 minute here. So, the cultivations of mushrooms is started some 1,000 years in China. So some books are given so many years is there. The many countries have been engaged which one there is of commercial production mushrooms productions. So they are becoming a different type of the technologies. Has been engaged which one is of commercial production mushrooms. The mushrooms production as the species generally example that is of agaricus, okay, bispose, and that is of in Wolverius species. So different type of the species is there. The mushroom cultivation is mostly indoor activity. The mushroom cultivation is not in outdoor, not a crab like structures. Always it is in only where the indoor sections. Just so we are using in only one room is enough. Indoor activity and is done in the farmhouses. Okay, the farm building should have so sterilization rooms along with the span. The seed we are saying that is of in span. So here now you see certain mushroom cultivations, mushrooms of cultivation. When you are cultivating the mushroom, this is all to the benefits. So nutrition values is there. Yes. So this is the steps of cultivations. Okay. This is the first. Now you see general steps of mushroom cultivations. The first now you see here pure cultures. Second that is of in span productions. Here span. So what is the meaning of span? Span means just like seed-like structure, seed. Okay. So we call that is of in span productions. And next now you see here. So next one preparation of substrate beds means so when you see first span means it is a seed-like structures. So next to preparations substrate of the beds like structures. Beds means one what we call that is a
Support, sir, on which it will hold.
Yes, yes. So that is a sub beds like structures is this. Just after when you are putting the span inside the beds, so after it is coming to like small, okay. So which one that is of span runnings. Okay, automatically the plant means the mushrooms is comes to outside that bed. So next one here it is in croppings. So here now you see here I'm saying first one that is of pure cultures. The mushrooms cultivated is carried out of the following manner. They select this to very easy and important in your examinations. So the writing part is very easy. Yes, now you see you write in only these steps is enough. Okay, these steps. What is the question here? Write the brief cultivation of button mushrooms. Is it clear? Write in brief cultivation of button mushrooms. You write these steps. What is the first one that is in? Please tell me here. Pure culture. Second one is?
Span production.
Yes, preparations. Then you write again after 1, 2, 3, 4, 5 completed. Now you see you write here the steps is important. How to prepare? What is the pure cultures? So what is the span? What is the preparation of the beds? Okay. So next to that is of span runnings and cropping. So next one that is of canning is this. Mushrooms for possible or to be consumed freshly if needed. They may be stored for a few days after canning detailed latter part. Okay, this is the one questions. And here now you see cultivation of agaricus and is called button mushrooms. Same type. This is also okay. Form design. Create all these points. Form design means just one room is there, one room. Same here, whatever you are completed before, now all these sections. The preparation of pure culture and span, preparation of compost, cupping and pausing, cropping and harvesting, the factors affecting button mushroom productions, harvesting, packing. First from design to the last that is of packing. So this is the process. Okay, so just you write 1 or 2 3 lines of what is form designs, how to prepare the cultures, how to prepare the compost. You know the compost, how to prepare compost. Compost means you are using to what we call that is in grasses. Okay, so in between the grass, put some water, some waters. So automatically, yes,
Compost manure, sir.
yes.
Yes, sir. We know, sir, that one.
Yes, these points and that is, yeah. So next now you see here, this is also one planning, making the mushroom compost, finishing the compost, cupping, yes, pausing, thinning and croppings. Okay. Yeah, here now you see this one, cultivation of agaricus and other things. See, this is the first farmhouse. Is it okay now? There will the farmhouse. Here now you see farm designs. This is the designs of the farms. Next one now you see here, this is a fin button mushrooms. Crop. Here now you see this is a how to prepare the compost. How to prepare these all the compost. So now you see here. So this is in also mushrooms are compost. So now you see here, this is in compost. Different type of the mushrooms is there here. Okay, 1 2 3 4 5 6 types of the mushrooms there. Okay, same here also. So different type of the mushrooms is there. Same form designing, raising the pure culture and span preparations, substrate and bed preparations, harvestings. Yeah, here now you see how to the grow. Yeah, first one that is often spores. So when the spores are produced to the hyphae, means so hyphae means it is a vegetative structures. One by one. After now you see the last here it is the mushrooms. Same. How to prepare the compost. Yeah. Already you know very well compost how to prepare it. So what we are using here? Natural compost and second there is often synthetic. Two type of the compost is there. So you know you see what is the natural compost, how you prepare the natural compost. And second one here you see now how to the prepare of synthetic compost here. Just you write in only general, that is also enough in this one. So harvesting and how to take the crop how to take here. Okay. So we are using different type of the methods here, harvesting. And now you see here the first flush of 3 to 5 days, and so here the compost where it is in the here this compost is there, and this one. Okay, first one that is often for the span preparation pure culture. Pure culture can be obtained of germinating the spores by where it is the growing pieces of inner tissues of mushrooms. Inner tissues of the mushrooms are a suitable that are sterilized culture mediums. Okay, So other button mushrooms are different type of the mushrooms is this. So here a crop cycle, the crop cycle minimum this is a 45 days, and 3 crops can be harvested from each bed. So daily watering of maintenance of 75%. You should maintain the minimum room temperatures. Okay. So that is often outdoor cultivation by using paddy straw. That is often cage cultures, cotton waste composing method, and polybag methods. The mushrooms are packed at egg state and are not allowed to grow to their maximum size and have shelf life about 6 to 8 hours at the room temperatures should be maintained. That is, they can be stored for a week and blazing to by air drawing. These mushrooms are affected by various weed, mold, and insect pests. So this is all to the mushroom cultivations. Okay. Here yes, see the growth requirements, raising the pure culture span preparations, substrate preparation and cultivation, mushrooms. So these are all the sections is there. So in this often there is a mushroom cultivation. Okay. Yes. So I next now you see here fungi. It is a biopesticides. Here now first one this is of in general fungi. Okay. So fungi it is a biopesticides. What is the meaning of in biopesticides? Tell me here anybody.
Like bio instead of chemical.
Biopesticides. What is the meaning of biopesticides?
Using living organism, killing of pests by using of living organisms.
By natural.
Eradication of pests by using
Parasitic
living
fungi live on derived groups of organisms, including animals and other microorganisms. Generally, the fungi, it is a 3 types is there. One, it is in based on the nutritional systems. One, it is in parasitic fungi. Second one, that is in saprophytic fungi. And third one, that is symbiotic. 3 types is based on the nutritions. Okay. Generally here, the parasitic fungi, it is living where it is in the group of organisms including animals and other microorganisms. Okay. Some soil fungi, the parasitic where it is nematodes, and these are referred to the nematophagous fungi, where the fungi is growing, where the nematodes above the fungi, above the nematodes. So we call that is a nematophagous fungi. So these fungi are being explored for the control and plant nematodes. See here, this is most important. Biopesticides. Pesticides means you know very well. Okay. But here, bio means life. Fungi, when growing above the nematodes, okay. What is going on here? That life cycle, that life is decreasing. Yes, nematodes life are decreasing when the fungi growing above the nematodes. Understand? That's why we are saying fungi, it is in biopesticides. Understand here? Mahati?
Yes, sir.
Please tell me why you are saying fungal, it is in biopesticides. Fungi is controlled to other lives. That's
Okay.
why we are saying biopesticides.
Okay.
Understand?
Yes.
Hmm?
Yes, sir.
There is a Mahapatra. Mahapatra, please respond.
Yes, sir.
Yes, what is why you are saying of biopesticides?
Because it can control the fungal diseases.
No.
Yes.
It control other life forms.
Yes, sir.
Other life forms. That's why that is in biopesticides.
Biopesticides. Yes, sir.
Biopesticides. Yes, that is important here only. Entire in this one.
Yes, sir. Yes, sir.
Okay.
Yes,
See,
sir.
when the fungi is growing above the life, above the one insects or anything, what happened here? The insects' life are decreasing. That's why you are saying it is a fungi, it is a biopesticide. Okay, then you can
Sir.
write any other things. This point is most important.
Yeah, yes, sir.
The fungi, you see here, the fungi grow insects, including the beneficial insects like silkworms. These fungi are called of entomogenous fungi. Such fungi are employed as a biocontrol agent in agriculturally important of insects, pests. Similarly, pathogenic weeds of crab plants, you see here, pathogenic seeds with crab plants, many soil fungi is there, such as trichoderma exhibiting the different phenomena like predations, competitions, antibiotics, and that's of parasitism inhabit the growth of other plant pathogenic fungi. So these fungi being developed in biocontrol agents for diseases. Different types of the fungal biocontrol agents are formulated with suitable carriers of it. So some are commercialized and sold market. Biocontrol agents are eco-friendly and specific to the target organisms. Is it understand? What are biopesticides? Give an account of fungal biopesticides. Understand here?
Yes, sir.
Okay. So here now you see some examples are given in your book. Please just you can go and read your there is a fungal, it is a biopesticides. So this is the fungi. And next one, what is the scope and techniques of fungal biotechnology? So what is the meaning of biotechnology? Biotechnology. Please tell me or anybody, what is the meaning of in biotechnology? Fungi, it is a eukaryotic organisms or prokaryotic organisms?
Technology used in biology, sir.
Biotech. It is the integration of natural science.
Biotechnology. See here, bio means life.
Life.
Technology. The technology are using by the life. We call that is in biotechnology.
Biotechnology.
It is in two types. That is already you know very well. Okay, we are not going the detail about in that one. The fungi, it is in biotechnology. Okay. Here now you see the fungi are eukaryotic organisms basically. So already you know very well eukaryotic organisms. So biotechnologically they are important group of microorganisms and contribute to industries, pharmaceutical compounds, production of, see, biotechnology. Once again, here I am telling, bio means life. This life we are using to the techniques. That's why we call it a biotechnology. That's why these fungus are using different type of the food industries. Fungus means the life. Yeah, one of the plant-like structures, a chlorophyll plant. These fungus we are using different type of the industries. That's why we are saying here fungus. It is a biotechnology. Okay, so the fungus we are using beverages, alcohol. Okay, so beside the severing, where the supplementary foods is there. So these fungus, they're also most important medicinal values is there. So medicinal importance also you are looking to the fungus. Where it is you are this fungus you are using so microbial proteins, the single cell proteins can be derived the fungi where it is in like the yeast. The mushrooms like yeast are used. You know the yeast?
Yes, sir.
Yes, the yeast we are using the food industries, bakery industries. It is produced to the. It is
Ethyl
produced to,
alcohol production also, sir.
yes. It is produce different type of the sugars. Okay, so that's why this fungus you are using different type of the food industries. The filamentous fungi, where it is a fusarium, so you also use it to the source of in mycoproteins. So here fungi, it is a biotechnology. The fungus we are using different type of the food industries and medicinals. So where the agricultural fungi that habitat of root zone, okay, where it is in mycorrhiza contributes towards the improving the soil fertility by way of mobilizing nutrients like different type of the phosphorus and so phosphorus where it is in a sulfur, where it is in copper and zinc, etc. Okay, so these are all the sections we are using to the fungus. The besides here improving of the soil physical properties like it is the aggregations and textures, etc. Okay. And the fungus, it is a biocontrol and biofertilizer agents. Accepted you are? The fungus, it is in biocontrol agents, biofertilizer agents, biofertilizers control, how to the controls. This is up to the control agents. Ashanti, please tell me, respond. Fungi, it is a biocontrol and biofertilizer agents. The soil fungi, yes. And you see the soil fungi are also serve biocontrol agents. So particularly where it is in penicillium and trichoderma and sporodesmis. Trichoderma species are an important organic matter decomposition of recycling of agricultural wastes. The fungi are good so deteriorating of biodegradation agents, and so we call that as of mycoherbicides. Mycoherbicides are known to control certain often where it's in some type of the weed controls, that the so other diseased seeds are controlled. This is also yes, the fungi produced the variety of industrially important compounds, as of in alcohol and organic acids, the vitamins, So next one, that is of in polysaccharides, and nucleotides are important primary, where it is in metabolitics. The secondary metabolitics include the steroids and alkaloids. There is often cyclopeptides. Cyclopeptides is there. So next one, that is often mycotoxins is there, and other one, that is in antibiotics is there. So next one, different type of the drugs is there. Fungicides are growth or promoting compounds. Okay. So next, you see when the cultivated fungi, so in the large scale on can adopt the fermentation purpose by using fermenters. The alcohols are produced by the yeast industrially using the fermentation technology. Where is the penicilliums? So already you know very well. Yeah. So there is a phylactum antibiotics produced by the fungi. These fungi are produced with the citric acid and is the major organic produced by where it is in aspergillus. Soil state for the fermentations, submerged fermentations are used for its productions. So means here, so the fungi, when you are using to the technology, here technology means so along with fungus, so we are growing. Okay, you see generally the fungi produced to the biomass productions. Okay, so the fungi source, it is a food and drugs. The fungi is a what we call it is in for the plant health. So next one, the fungi, different type of the industries is there. So fungi, it is a biocontrol agents. How the biocontrol agents means? For the fungi, we are using sewage treatments. So when you see here, fungi is growing in this area. So when I'm putting the water, some concentrations, okay, the fungi is absorbing. That's why we are saying control agents. Okay, sewage treatments steps is there. One, two, three. Here one fungus is there. Fungi, 3 steps is there. So when I am putting the water, the wastewater, you see the Hussain Sagar. You said the Hussain Sagar, we are doing sewage treatment. The sewage treatment, so they are germinated. One the fungi. These fungi is absorbing some toxins, some all other things. That's why when the water is coming in this area, okay, it is absorbing all that these minerals, some other things. That's why the fungi is a biocontrol agents. Yeah. So this is after the, so fungi, it is a biodegrading agents. So next, now you see here the techniques that is of in see, so where the starter culture for inoculum production is there. Second is of fermentation methods. So already you know very well fermentation method means fungi we are using. The fermentation, their applications is there. So next, now you see here different type of the fermentation is there. Fermentation and the process is there. The fermentation methods is there. The biomass production of the fungi. So here, this is of fungi biotechnology means the fungus we are using with the different type of the in other life forms. That's why we call here it is in fungi, it is a biotechnology. Yes, so now you see the fungal enzymes and metabolitics. The fungus are source of food, organic acids, that is of in already I told in the alcoholic industries. The fungi produce a different type of the enzymes. The fungi, it is a growth promoting substances. It is a where it is in the amino acids, etc. So the recent techniques of protoplasm fusion of recombinant DNA technology, we are also using in the DNA technology. So the role of fungi, their action of understanding with the biotechnology process. So that is of industrial products like organic acids and alcoholic enzymes. Similarly, second one, second there is of pharmaceutical products like where it is in antibiotics, so microbial conservations, and next one that is of in food and food stuff products where it is in SCP, means the fungi we are using in all the sections, entire the sections we are using. The fungi, it is a main source food. Okay, the fungi, it is produce organic acids. The fungi, it is produce the alcohol. It is produce enzymes, growth promotings, and it is produce amino acids. So that's why the fungi, it is a main domain. We are using different type of the industries. Okay, so the biotechnological scientists have so discovered that foreign genes put filamentous fungi, so then use it to the desired of the products. Some filaments like fungi, that is of penicillium and aspergillus. Already you know very well. The penicillium is the famous one, substance is the antibiotic. So means we are using different type of the industries. The fungal enzymes. So here the main enzyme, that is of amylase enzymes. The enzymes. So before going that one, you see what is the meaning of enzymes? Enzymes means the reaction is going very speed. We call that is of enzymes. The enzymes are biological catalyzed by the biochemical reactions leading to the microbial growth, where the in all the metabolic reactions, respirations, in wall formations, fermentation products. The total world market for the enzymes in 1985, so we are using to 1 lakh more than 50,000, 1,50,000 tons are enzymes we are using. The enzymes are used distinct fields, so that is with the gene manipulations, analytical agents, industrial of catalyst. Enzymes are produced annually. The most hydrolytic enzymes, where it is in 80%, so 60% of protease enzymes. Enzymes are extracellular and intracellular types. Mainly amylase enzymes, amylase, and second one that is of where the cellulosic enzymes and lactase enzymes. That is of next one pectinase enzymes. So next one that is of proteaceous, proteaceous enzymes. Means the fungi is produced different type of the enzymes, different type of the enzymes. These enzymes are using different type of the techniques. Okay, so the fungi are enzymes. So you see here where the manufacturing companies is in that area. So we are using to amylase enzymes. So and now you see here the vinegar. So the cellulose enzymes, so where you have preparation to the vinegar enzymes, and the lactase enzymes, the processing of the milk lactase enzymes, but the milk process industries we are using in lactase enzymes, and so other one that is often where the fruit juice industries. So there you are using of the pectinase enzymes. So that is often another one. So where the bacterial industries they are using to the protease enzymes. So here the fungi it is produced to different type of the enzymes. So we are using a different type of the enzymes. And next now you see the fungus are produced where the vitamins. So different type of the vitamins are produced to the fungi. So all the phototrophic microbes are capable synthesizing vitamins for their growth. For their growth, vitamins are required in trace elements. Means the fungus are produced different type of the vitamins and proteins. So here one keeping the here it is in it is of microproteins is there. So means different type of the proteins are produced to fungi and some other organic acids. So in that organic acids already you know very well in the citric acid, gluconic acid, fumanic acid, and teponic acid, ozic acid, lactic acid. So gallic acid, malic acid, other organic acids. So here the fungi are produced different type of the acids. So we are using different type of the industries. And some of the fungi are produced the amino acids. That is a tryptophan amino acid is there, and so next one that is in lysine amino acid is there, and so that is of in methionine amino acids is there. The world production, the amino acids have exceeded 200. And that is of in supplements, feeds, and food. The chemical synthesis, enzymatic transformations, or the techniques used to different type of the industries. Okay. Next one that is of in lipids. So lipids, the fungi are produced with the lipids, and next one, the fungi is produced with the even alcohol also. Okay. So you see, India is the rich. Our country is the rich fungal industry is there. The several fungus, the species are much important in the agriculture, medicine, and with the food processing industries, and next there is on beverages industries. Most common fungi such as aspergillus elaborates around it is produced to generally 20 types of enzymes, 20 types of enzymes. So which are important and industries. Similarly, so other trichoderma like that fungi. And see where the cellulose enzymes, so it's the most commercial values, and already you know very well single cell proteins become the important production fungi. Okay, so that is an amylase. So next one that is often proteases, pectinase, liganase. So all these enzymes are using. Okay, so different type of the chemical industries. Next, so the fungi produce a different type of the organic acids. So these organic acids are very commercial values is there, commercial production of organic acid by the microbial other fungi, limited member of gem that is of mainly so aspergillus. The organic acid produced from the fungi, gluconic acid. So these gluconic acid also we are using so very much medicinal industries. Okay, so this is the fungi, fungal enzymes and metabolites. So once again, see the fungi is produced different type of the enzymes. So that is the amylase enzymes, cellulose enzymes, lactase enzymes, and for next one is of pectinase enzymes. So and other one that is in proteaceous enzymes. Okay. Similarly, the fungi also produce a different type of the vitamins. So already you know very well what is vitamins. So why you are using to the vitamins, and so where the proteins are also produced with the fungi and other organic acids. Okay, so amino acids, lipids, and alcohols. So this is uh, the fungi, enzymes, and metabolites. So here now you see write about the fungal enzymes. Okay, so next you see here. So give an account of organic acid produced by the microbes. And here now you see the fungi is produced different type of the organic acids and enzymes, proteins, and vitamins. Entire all these sections, here one is most important is there industrial production of penicillium and citric acids, penicillium and citric acid and alcohol. Okay, production of antibiotics. Already you know very well the penicillium. Okay, the penicillium it is in produced. Okay, antibiotic, the famous antibiotic you know very well. Production of antibiotics. The penicillium it is a saprophytic or a parasitic?
Saprophytic, sir.
Saprophytic. Yeah, the fungi. Just 1 minute here. I'm showing one more for this PowerPoint. So here it is. Files. So now here, industrial production. Visible here. Industrial production, penicillin, citric acid, and please respond. Anyone? Mahapatra.
Yes, sir.
Here, this is all to the objectives. Is there? Rid of these objectives here. This one's penicillium. Okay. Penicillium it is a bio reactors was used to grow in penicillium mold. The fermenters. Here now you see this one is most important name is there by the Alexander Fleming. Okay, sorting petri dishes of containing the colonies of Staphylococcus. He noticed that plate was rooted to the colonies. The zone around mold was later identified as the rare strain Penicillium genus. So here this stuff in Penicillium. So deep fermentation methods, Penicillium productions. Okay. The penicillium we are using in antibiotics. Okay, so here two process sections is there. The one that is of in upstream processing, second one that is in downstream processings. How the production of penicillium. How many types of process is there? Two types of process is there, upstream processing and second one that is of in downstream processing. So now you see what is upstream processing. So very simple here. The comprise of any technology that leads to synthesis of product and includes exploration, development, and production. So we call this stuff in upstream. Similarly, second one, downstream processing, comprising extraction and purification of technological product for fermentations. Two types of methods is there. So now you see how the prepare of preparation to the penicilliums. So medium for reproduction system, medium for productions. There is a carbon source is there, and next there is a heat sterilizations. Two types is this. Okay, how to the production of. This is all to the fermentation methods. Okay, where the next one is of some somebody is asking when we use the microorganisms for any useful purpose. So microorganisms, so we are using in all the industries, particularly not only one sections. See, for example, penicillium. Okay, so now we are studying in the fungus, microorganisms. So this fungus we are using different type of the industries, already told, like beverages industries, alcoholic industries, okay, so and bakery industries, different type of the industries. So when we are using in that fungi, the fungus is produced to different type of the chemicals is there, alkaloids is there. That's why, so the microorganisms are using in all the industries, not only particularly one. Some of the microorganisms we are using by the preparation the medicines. Some of the microorganisms are using where the medicinal industries. Okay, different type of the industries we are using. So this is the processing methods. Okay. So these are in extraction methods. Characteristics of the penicillium. Already you know very well the penicillium. The penicillium belongs to which group? That is of in Zygomycotina or Ascomycotina. Which group is penicillium?
Ascomycotina.
Very good, Ascomycotina group. Now you see this one, it is a penicillium. Visible the penicillium. Here is a vegetative hyphae is there. Conidiophores is there. Okay. So this of in Metuda and this of in Conidiums is there. So monoverticillate and biverticillate, and here this of in multiverticillate. But here no need all these. What is the life cycle in this one? But here only important. The life cycle we are already coming in the first year. But here we are using the preparation. How to prepare the penicillium? Okay, what is the process? The penicillium we are using to the antibiotics. So how the prepare of antibiotics, that is important. The first production, how the production of penicilliums. Okay, so what is the production process? So that one is this here is important. But what is the life cycles? What is the root system? What is the stem system? So what is the mycelium? No need here. Sir, here only. Sir, this penicillium we are using. Okay, so by the antibiotic purpose. So when you are using an antibiotic, the process is this. So just to write in only that process. Yeah. Here now you see this is a production of penicilliums. Here this is most important. Okay. So media ingredients. Okay, now you see here step by step is there. Preparation of incubation stems, preparation of media, how the prepare of penicillium. Okay, the penicillium always it is developed were the saprophytic. Okay, but now here the penicillium are development in my laboratory, in our laboratories. So just you used to these methods, this type of the production of penicillium methods is there. Yeah. So this is of in process. So this is of in aerobic respirations. Aerobic. The whole process we call that is in aerobic. Okay. But now we are not going to detail about in the aerobic. So aerobic is other part, but you write in only in the process of and the mechanism of development penicillium. So that is of an aerobic. Okay. The fermentation is carried of 4 to 5 days and at 25 to 27 degrees centigrades. This gives a yield of, you see now here only just about 2025% of penicilliums. Yeah. So this is of in process is there and production of the formations. The penicillium is produced secondary metabolites. Here, what is the meaning of metabolites? Whatever you are, see the penicillium are produced to different type of acids. So the acids we call that is in metabolic. Certain species of the penicillium is produced when growth of the fungi inhabited stress. It is not produced during the active growth. Okay, so this of methods is there, and here production of organic acids. Similarly, same. So the organic acids. So the fungi, they play important role in the production of alcohol, organic acid, antibiotics, and other enzymes, vitamins. Okay. So now you see, just you can go and throw your book. What type of alcohol is there? What type of organics? What type of antibiotics and enzymes? So I already told. Just once you read, this is totally general. It's totally general. Vitamins, you know already. A, B, C, D, and K vitamins means the fungus are producing all types of vitamins. The alcohol produced by the yeast of fermentation technology and ease of importance of in alcoholic productions. Antibiotics like already you know that one, the penicillium. So the lactin group of production of penicilliums. The major production of citric acid, a composite where it is in citric acid. Citric acid is most important. So here now you see aspergillus. Aspergillus is produced to citric acid. Okay. So like not only citric acid, now you see here gluconic acid, malic acid. Okay. So and butaconic acid. Means these are all the produced, which aspergillus are produced these type of the acids. In this one, the citric acid means the citric acid is the most important. So in our industries, that's why the aspergillus we are using. So now you see here, this is a different type of the fungi of microorganisms. Aspergillus, different type of the aspergillus is this. Okay. So you know you see this is of yeast is this and yeast. So this is of production of citric acid. This in examination purpose, this one is important. Surface of fermentation process, submerged fermentations, soil state fermentations. We call this of Koji process is this. Koji process. So here preparation of solvent extractions is this. Of fermentation methods and other raw material to alcoholic production. This is a anti relation to the pollutions. Okay, so this is also one most important topic is there. So this one we will discuss in the next classes. So any doubt?
No, sir.
Yes. See, whatever you are completed is totally in the just like general types is there, and in spite of just once you read, that is enough in this one. Okay. So here the fungi general accounts. So in this one, the fungi general account means what type of reproduction system is there? What type of reproduction system? So when giving these questions, reproduction is 3 types. So what type of reproduction systems? So that is asexual is there, sexual is there. Okay. So vegetative, it is a common vegetative. So and in this one, parasexuals. What is the meaning of in parasexuals? So fungal taxonomy. So you know very well already. Means Ainsworth classification is most important. Ainsworth classifications. Okay. So and next one is a mycorrhiza. Okay. Discuss importance of mycorrhiza in agriculture and forestry. Please tell me any one point. Mycorrhiza. What is the use of mycorrhiza?
Phosphorus.
Very good.
Phosphorus.
Yes. But when it is coming to examination, just expands. You write little bit.
Endomycorrhiza, ectomycorrhiza.
That is other question. Here, okay, the mycorrhiza are produced. Mycorrhiza are produced different type of the chemicals. That chemicals are very used to a higher plant. Okay. So based on this point, just can expand your answer. Understand?
Sir, chemicals means enzymes, sir?
Enzymes or anything.
Okay.
All other things, not only one.
Okay.
The fungi are, yes, fungal are produced different type of the chemicals. These chemicals are using where the plant. Just one, when you are writing in the one word, please expands that sentences. Is it okay? Similarly, mushroom cultivations. So it is also very simple and very general questions. Okay, mushroom cultivations. So important. What is the scope of in biotechnology? When the fungus are using, okay, different type of the industries. Okay. That is of in today's, yeah. So we will go to the next one. So any doubts, anytime, can ask to me. Is it okay now?
Okay,
Can I
sir.
leave?
Okay, sir.
Okay.
నమస్కారం సార్, నేను హనుమంతరావును సార్. సార్ వినపడుతుందా సార్?
Yes. Tell me.
థ్యాంక్స్ సార్, చాలా బాగా చక్కగా ఉంది క్లాసు. కొంచెం కొన్ని క్వశ్చన్లు ఇంపార్టెంట్ చెప్పడం ప్లస్ క్లాస్ కొంచెం బాగా చక్కగా ఉంది.
Very good, nice.
చేతికి ఏదో అయినట్టుంది సార్, మీకు కట్టు కట్టినట్టు అనిపించింది.
Yes. Little bit problem.
సార్
Air crack, little air crack.
అరే సరే సార్, థ్యాంక్ యూ.
Okay. Get out. Karthikeyan Ramanathan.